Something I want you guys to understand is in mammalian reproductive systems, essentially, and this includes humans, in early embryonic development, everybody has the same reproductive structures. You have the potential for either male or female gender. It depends on presence of a certain gene and concentrations of different hormones to determine which direction development occurs in. But relatively early on, it is differentiated as to whether the embryo is going to be male or female. In male reproductive system, what we're going to do is we'll look at a little bit of gross anatomy, and then we'll turn around and deal with this in terms of what's going on in the gonads and transport of the semen out of the body. So, a few basic concepts. Those of you who haven't had lab yet, there are other details we'll be dealing with in lab in terms of structural arrangement. But, looking at male reproductive system, a couple of things we need to know about right up front here. First off, the penis is going to have running through it the urethra. And you have columns of tissue in the penis that make up what they call the erectile tissue. There are two different types. There's corpus cavernosum here and corpus spongiosum here. All right, gang. The erectile tissue is a series of blood sinuses with arterioles feeding into them. The arterioles have muscular walls. We know that from cardiovascular system. They're usually going to be somewhat constricted. Interestingly enough, under parasympathetic stimulation, they relax and dilate and allow more blood to pass through all of these little sinus-like cavities. Around these cavities, we have elastic connective tissue. What ends up happening is more blood flows in than can go out. As these cavities fill and become turgid, erection occurs. The corpus spongiosum, you notice, is not subdivided like we see in the corpus cavernosum. Corpus cavernosum, we have additional connective tissue partitions forming chambers. So this allows for higher pressure. We can't do that in the corpus spongiosum because it's surrounding the urethra, which is where the semen is going to be passing through. If we compress the urethra too much, we're not going to be able to move semen through it properly. This is the reason for the two different kinds of erectile tissue. The gonad itself, the testis, is going to be located here in an integumentary sac we call the scrotum. All right, again, fairly early on, testes start developing up in the body cavity, and they're going to descend over the anterior aspects of the pubic bone into this integumentary sac. Reason for this is core body temperature is too warm. Spermatozoa, the male gametes, are temperature sensitive. The core body temperature is too high. They tend to die off relatively quickly as a result. So by having the testes descend down into this scrotum, you're getting outside of the body core, and you literally are dealing with temperatures a couple of degrees cooler. That is the whole point of this arrangement. Female gametes don't have this issue. Male gametes do. Now, what we're going to do is we're going to have a look at what goes on in the testis and work our way back through the system. So, with respect to that, if we take a look at a sectional view, if we take a look at a sectional view of the testis, you're seeing essentially a transverse section here. You note it's divided up into a series of lobules. You've got connective tissue septa separating these. And within each of these lobules, you're going to have essentially highly convoluted tubes, and they call these seminiferous tubules. These are where spermatogenesis is going to occur. This is where sperm production is going to happen, in these seminiferous tubules. We'll get into the process here in a moment. But as the spermatozoa are produced, they're going to be moved from the seminiferous tubules into what they call a straight tubule. And from the straight tubule to the reedy testes. Reedy testes are essentially a net-like series of passageways. And the reedy testes are going to carry the spermatozoa by way of the efferent ductules into a structure that wraps around the testicle that we refer to as the epididymis. The epididymis is going to store the sperm. Now, again, spermatozoa are very, very small, and each one has a flagellum. They are not mobile at this point. All the way through this, the spermatozoa are not capable of moving themselves. They don't get activated until immediately before ejaculation. So all we're going to do in that epididymis is basically store them and provide them with nutrients, support them. They don't have enough cytoplasm to store any significant amount of nutrients. All right, production of the spermatozoa in those seminiferous tubules. Basically, it's going to be done by way of a process that we call spermatogenesis. All right, gang, y'all remember when we talked about cell cycle? We talked about mitosis. Yeah. During spermatogenesis, we have another process very similar to this, another kind of nuclear division that they call meiosis. The way it works, just giving you the bare bones of it, because we could spend almost the whole class period talking about this process. Meiosis differs from mitosis in that there are two separate divisions that we call meiosis 1 and meiosis 2. Instead of producing 2 daughter nuclei, by the end of meiosis 2, we have 4 daughter nuclei. Now, you all have 46 chromosomes. 23 came from your mom, 23 came from your dad. We have then pairs of what they call homologous chromosomes. We're going to separate homologs during meiosis, but they don't all separate in any defined pattern. Each gamete has some chromosomes that came from your mother and some from your father, but they're not all going to have the same combinations. This appears to be pretty much random. You're going to get one homolog out of each pair, that's guaranteed, but you don't know which homolog out of each given pair you're going to get. So we have an increase in genetic diversity that occurs here. Basically, our primary spermatocyte, both of these chromosomes are present out of each homolog. It has what they call then a diploid chromosome number. That would be 46. What we're going to do with meiosis 1 is we're going to cause the nucleus to divide in such a way that only one homolog out of each homologous pair ends up in each daughter nucleus. The result, each of those secondary spermatocytes at the end of meiosis 1 only has 23 chromosomes, 1/2 the normal number. These are the only cells in the male body that are going to be like this. The second meiotic division is just mitosis in each of those nuclei. So we end up with 4 haploid nuclei. And as cytokinesis occurs, then 4 haploid cells. As a result, these spermatids are then going to essentially kind of degenerate into the actual spermatozoa. Now, spermatozoan, with a high power objective on your microscopes up in the lab, they'd be barely visible. They're incredibly tiny. A typical spermatozoan is going to work something like this. We have our spermatid, and it's going to develop a flagellum, and it's going to start losing cytoplasm. In fact, it loses almost all of its cytoplasm, most of its membrane, until gradually it's reduced down to just a tiny little bit of cytoplasm around the nucleus of the cell. And here, in what they call the middle piece, we'll have the remaining mitochondria providing energy to move the flagellum. And the neck piece is going to have the centrioles that are left. And that's really almost it. At the very tip of the head, there is a big vacuole that they call the acrosome, or acrosomal cap. And basically it carries a particular kind of digestive enzymes. And that's really all there is to the spermatozoa. Gang, these are produced in huge numbers. They are fairly cheap to produce. And in the average lifetime of any given male human, something on the general order of 99.999% of them are going to die. There's only a tiny percentage of them that ever actually end up being used in fertilization. By the time they reach the finished spermatozoan stage, they'll have been moved into the epididymis. The flagellum is inactive at this point. Being stored in the epididymis, basically what's going to happen is some, not all of them that are in the epididymis, but some, with the nutrient solution that they're suspended in, will be moved toward the penis when ejaculation is imminent. So you have arousal occur, which causes filling of the erectile tissues, corpus cavernosum. There are two bands of that, one band of corpus spongiosum. And as ejaculation is imminent, and here I have to go back to a previous illustration, as ejaculation is imminent, we're going to take and move some of the spermatozoa from the tail of the epididymis here. You've got them all through this highly convoluted tubule in the epididymis, from the head all the way down to the tail. From the tail, we're going to get some of the spermatozoa in their nutrient solution forced into this tube here that we call the ductus deferens. Now, how are they going to be moved in there? Mainly it's by smooth muscle in the wall of this structure going through peristaltic contractions. We talked about peristalsis in digestive system. Peristaltic contraction is going to be moving these. It's going to move through the ductus deferens and basically pass on up into the body cavity. Now, going back to our sagittal section, because that's going to be the best one to show this. All right. So peristaltic waves are going to carry the spermatozoa up over the superior anterior aspect of the pubic bone, over the superior aspect of the urinary bladder, medial to the ureter, and then it's going to come back down again. We're only spending a very brief amount of time in the body cavity here. And here we have what they call a seminal gland. Seminal glands also, in some of the older books, are called seminal vesicles. In male reproductive system, the material that they call semen is not mainly sperm. Fact of the matter is, only 5% of the volume of the semen would be spermatozoa and the nutrient solution from the epididymis. That means 95% of it is going to be other materials. The seminal gland produces 60% of the volume of the semen. So, this glandular structure here is going to introduce fluids into the end of the ductus deferens as the spermatozoa pass by. It mixes up with the nutrient solution around the spermatozoa. The duct that is formed here, going through the prostate gland, is what they call the ejaculatory duct. All right, gang. Secretions of the seminal gland. There's going to be a large percentage of this is going to be a kind of sugar. It's mainly fructose. That's going to serve as an energy supply for the spermatozoa. There's also going to be some enzymes incorporated into it. And actually, between here and the prostate, we're going to activate the flagella. The spermatozoa have not been capable of moving themselves around until this point. Prostate is kind of an almost ball-shaped gland that encompasses the urethra and the ejaculatory duct. And the prostate is going to add about another 30% to the volume of the semen. Okay, now we've got 95% of the volume accounted for. Materials from the prostate gland, there's some of them they haven't identified yet. But among the ones that they know include things like, I don't know, how about citric acid? There appear to be some materials here that act almost like an antibiotic. There are materials in here that act as coagulating agents. They cause clotting of the semen itself. This is going to form essentially a semen plug after ejaculation occurs to help keep the semen in the vaginal cavity. It's a temporary thing, but again, it's necessary for the process. So as we pass through the prostate, we're adding these materials, and the urethra here is what they call the prostatic urethra, where the ejaculatory duct meets the prostatic urethra. We form what they call the membranous urethra, tiny little segment right here between the prostate and the base of the penis. Once we pass into the base of the penis, it becomes the spongy urethra because it's running through that corpus spongiosum. But we've got one more gland here that we have to deal with. And that gland is what they call the bulbourethral gland. Bulbourethral glands, the old name would be Cowper's gland. Bulbourethral glands are relatively small glands. There's two of them, one to each side of the midline. They produce about 5% of the semen volume, and it's actually released ahead of the rest of the semen. There's going to be a fair little bit of mucus in it to help lubricate the rest of the urethra. And it also has some things that they call buffers. We talked a little bit about buffers and digestive system, yes? One of the things you have to keep in mind is normal urine has a fairly acidic pH, or a slightly acidic pH. Spermatozoa don't do well in an acidic pH. They actually need a somewhat alkaline pH. So what this does is it buffers any acids left in the spongy urethra to essentially prolong the life of the spermatozoa. Now, the spermatozoa are being moved mainly by peristaltic contractions in the walls of the urethra, all the way out through the external urethral orifice. This is what happens during ejaculation. This is during coitus, then the semen gets deposited into the vaginal cavity. Part of it clots. This essentially keeps it in the vaginal cavity. And the rest of it, the to the point where ejaculation is occurring. Now we need to look and see what's happening on the female side. Female reproductive system. Basically, as we take a look at it, okay. We're going to start with the ovary, and again, kind of work our way back. The ovary, there's two of these, and they are the female gonads. Yes, they're going to be what's responsible for producing what are commonly called ova. Now, gang, when they describe this in your text, something I need to point out to you all is that oogenesis, production, the process of producing ova, there's some changes that are coming about that haven't made it into the textbooks yet. A couple of years ago, helping brainstorm a new physiology text with one of the publishers, there were like 14 physiology instructors from around the country that were involved in this. And 3 of them were doing active research in reproductive physiology. And some of the research that they've been doing indicates there's some changes in how we interpret oogenesis as it occurs in human female reproductive system. What you see up here and what's in your book is pretty much the standard line at present. Oogenesis actually begins before birth. Spermatogenesis typically doesn't begin until puberty. Okay, there's one big difference right there. Production of the primary oocyte, which is diploid, happens before the individual is born. Now, those of you that have had, maybe in high school or something, some anatomy and physiology of some kind, may have heard that your females are born with all of the primary oocytes they're ever going to have. Most current research is indicating this may not be the case. There may be some production of primary oocytes that goes on pretty much throughout the reproductive part of the lifetime. How much is something that's still open to question. But they're now seeing evidence that this idea may not be 100% correct. So some of the current research that's being done is indicating that what you're seeing up here may be changing in textbooks in the next few years. And this is the way it's been in textbooks for decades. All right, in any event, these primary oocytes, most of them are produced prior to birth. That much is still going to be true. Meiosis I is going to be completed by puberty. Okay, we've got a significantly longer process going on here. Meiosis, the whole process of meiosis to produce those spermatids takes a couple of days to get to the spermatid stage. We're talking about primary oocytes being produced before birth and meiosis I occurring and finishing at about puberty. We're talking years here just to get through the first division. The second division of meiosis may never be completed. The second meiotic division only occurs as a result of fertilization. The vast majority of these oocytes never come in contact with the spermatozoa, which means in most cases, meiosis II never happens, or is never completed. Also take note, meiosis I, we've got a secondary oocyte that's produced as a result of that, and something they call a polar body. The secondary oocyte is the functional oocyte, and it's large. Gang, this oocyte is almost the size of a period at the end of one of the sentences in your textbook. It's huge. It has a fairly substantial nutrient storage capability. That polar body, they don't know if they do anything at all. Right now, they're considered non-functional. If meiosis II happens, you're going to get another polar body, a second polar body produced, and one functional gamete. From one primary spermatocyte, we got 4 functional spermatozoa. From one primary oocyte, we're going to get 1 functional mature female gamete. Female gametes do not have any kind of locomotory structures at all. They just kind of drift. They are substantially larger than the male gametes. They're produced in far fewer numbers, like several orders of magnitude smaller numbers. So there's a pretty substantial difference in gamete production as it occurs here. Maturation of these oocytes, secondary oocytes, is going to occur like so. If we take, just a moment here, if we take and look at this, when we talked about anterior pituitary gland, I mentioned to you guys a family of hormones were called gonadotropins. Yes? Okay, 2 of these are going to come into play here. Basically, we have follicles. These would be clusters of cells around these secondary oocytes. And what they call the primary follicles are going to be relatively small. We're going to have an increase in what the FSH there stands for, oddly enough, follicle stimulating hormone, produced by the pituitary. We get an increase in production of follicle stimulating hormone. And that's going to cause some of the primary follicles to mature. In the ovary, there are clusters of primary follicles. They actually call these nests, where you've got many, many primary follicles. At this point, they don't know how the selection process works, but they do know that multiple follicles will begin maturing. And by maturing, I mean they start increasing in size. The tissues around the secondary oocyte, you get more mitosis and cytokinesis, you get bigger masses of tissue, and you start developing what they would call secondary follicles. Secondary follicles would be like this and this. Only one, under normal circumstances, is actually going to be involved in releasing an oocyte. Basically, the tissues around the oocyte also start producing hormones to help support this process. The first one to become self-sustaining, if you will, is the one that survives. The rest of them actually die and are broken down. So for every one secondary oocyte that's released, there are several that die and are destroyed. And this, again, has been confirmed even with most recent research. Only one is going to get to the level of what we call a tertiary follicle. Now, follicle-stimulating hormone concentrations start dropping, and we're going to see an increase in release of luteinizing hormone. But there's another player here, and that's estrogen. What we're getting produced here is a group of hormones that they call estrogens. Estrogens essentially are going to some small degree, inhibit production of follicle-stimulating hormone. And as they peak out, they're going to stimulate production of luteinizing hormone. This is what's going to cause ovulation to occur. This big peak in estrogen here here, and the big P and luteinizing hormone that we see up here. Those two events coinciding are what are going to initiate ovulation. Now, for this, we need to go back and have a look at our ovary. So, throughout this process, our primordial follicles here are going to start developing. By the time you get to the secondary follicle, virtually all of them have died off but one that moves on to the tertiary follicle. And by the way, the tertiary follicle has a large mass of fluid in it. It's like a giant vacuum. Tertiary follicle, as we get that peak in estrogen and luteinizing hormone, is going to move to the medial side, the medial end of the ovary, come in contact with the ovary wall, and literally rupture. It bursts, and as it does, the fluid in this cavity here is dumped out. And with it, we have the secondary oocytes, and around that, just a little bit of this tissue from the follicle. And that would be at that point referred to as a corona radiata around our secondary oocyte. Follicle's not done here. The follicle, remember, is generating estrogens. The empty follicle is then converted into entirely a temporary endocrine structure that we call corpus luteum. And it's going to continue producing estrogens and some progesterone for a short time. It is not going to last forever. It gradually starts atrophying, getting smaller. Hormone production drops off, and eventually it condenses down into a tight mass they call the corpus albicans. At that point, it's no longer actively producing hormones, and it's eventually going to be broken down and reabsorbed. Okay, what else have we got happening here? Well, for that, we have to look at the system overall. Okay. In the female reproductive system, you've got two lip-like structures that they refer to as labia externally. Labia majus and minus. A little bit of erectile tissue. This is erectile tissue very much like the corpus cavernosum in male reproductive system that they refer to as the clitoris. And then take note, the urethra here is not part of the reproductive system. Unlike the male reproductive system, we do not have the urethra being used as a common pathway. Instead, we have the vagina. This is where sperm would be released during coitus. And here we have the uterus. Uterus has three primary layers: paramecium, which is the outer covering, the myometrium, which is this thick muscular wall here, and the endometrium, shown in white here. The endometrium is variably thick. It's going to be a combination of epithelial and connective tissues. It's highly vascularized. And one of the things that happens is as luteinizing hormones concentrations increase, as well as estrogen and progesterone, one of the things we see happening is tremendous proliferation of cells in the endometrium. The endometrium literally becomes much thicker in terms of, to the point of being several times its ordinary thickness. You get an increase in vascularity, so it becomes even more highly vascularized. And this is all in preparation for implantation of an embryo. All right? So this is happening, this is really starting long about the time ovulation would occur. In dealing typically going to occur on the medial side of the ovary, I'm sorry, on the lateral side, pardon me, of the ovary. It's going to occur out here. And take note, coming off the uterus here, we have two uterine tubes, one going to each ovary. Uterine tube, the old name for it was fallopian tube. Uterine tube goes out laterally and then curves back medially. And at that curve, it expands. The expanded portion here is what they call the infundibulum. The infundibulum has projecting off of it little finger-like projections that they call fimbria. All right, gang. My left fist is representing lateral end of the ovary. My right hand, the hand being the infundibulum, my fingers being the fimbria. The fimbria literally wrap around the lateral end of the ovary. Now, they're not holding it in place. There are other aspects of connective tissue that do that. What the fimbria are doing is this. On the side toward the ovary, the epithelium is ciliated. And in fact, all along the uterine tube, you have ciliated epithelium. It's a fluid-filled tube. When the secondary oocyte, with that fluid from that chamber in the tertiary follicle, is dumped out, the cilia create a current that draws it into the infundibulum. And it's just going to drift through the length of the uterine tube. Under ordinary circumstances, ovulation is going to happen in one ovary at a time. Now, which ovary is selected and how, again, they're still working on it. They're still trying to figure that out. That is not the only way it can happen. There are women who can have multiple ovulations occur virtually at the same time. That's actually more common today with treatments for fertility issues than what it has been. But there are people without any fertility treatments at all that can have this happen. Yes, ma'am?
I'm sorry, where did you say that this ciliated epithelial is?
It begins on the side of the fimbria closest to the ovary, and it runs all the way through the length of that uterine tube.
So it's inside the uterine.
It's inside the uterine tube. It's going to be used not only to draw the secondary oocyte in, but also to propel it through the uterine tube. We've got to keep it moving, and it doesn't have any way to move on its own. All right? Now, assuming coitus has occurred and ejaculation has happened, the rounded projection of the uterus projects into the vagina. It's called the cervix. It has an opening in it that they call the external os of the cervix. Spermatozoa are going to move up through this opening into the uterus, and they're going to follow the uterine lining on up to the uterine tubes. Okay, here's another place where they've got a big question mark in human reproduction. How do they know what tube to go into? They don't have eyes. Okay? In other aspects of biology, there is some precedent for the oocyte releasing some kind of chemical that acts as kind of a trail for male gametes to follow. That has been identified in other kinds of living organisms. It may be that human secondary oocytes do the same thing. We don't know for sure yet. If so, it's present in very minute quantities, which makes it even harder to identify. We do know that some spermatozoa end up in both uterine tubes. The ones that go on the wrong one, of course, die. You have to understand, the female reproductive system is inherently hostile to male gametes. First off, it's up in the body core. It's several degrees too warm. Secondly, the normal pH of the female reproductive system is slightly acidic, and spermatozoa don't do well in acidic circumstances. So these spermatozoa start dying off fairly quickly. You can think of it as kind of a selection process. Those that have some kind of an issue with their flagella and things of this sort don't make it. The ones ones that end up in the appropriate uterine tube, when they encounter the secondary oocyte, it's still got that corona radiata around it. Okay, here's where the acrosome comes in. That acrosomal cap containing the special digestive enzymes, literally it starts digesting through the corona radiata to the secondary oocytes. The part of the spermatozoon that actually penetrates the outer membrane of the secondary oocyte is just the head. That has the nucleus. Okay, think back to way back at the beginning of the semester when we talked about cell structure and function. I pointed out to you that for the most part, all of your mitochondria come from mom. Here's how that happens. All the mitochondria of the spermatozoa are in the middle piece, the part that doesn't penetrate the secondary oocytes. As soon as the head of the spermatozoon penetrates the outer membrane of the secondary oocyte, that triggers meiosis II, the second meiotic division. You only get one functional nucleus out of that division. That nucleus is going to be haploid. It's going to have 23 chromosomes. The spermatozoan nucleus is haploid. It has 23 chromosomes. They fuse to form a zygote. Okay, timing here is important. If this happens either in the infundibulum or the part of the uterine tube closest to the infundibulum, you've got a good chance of a pregnancy resulting. Fertilization of that oocyte does not result in a pregnancy. That is not what a pregnancy is. Fertilization of that oocyte does not in any way by itself guarantee that pregnancy is going to happen. What's going to happen here is this. The first diploid cell produced is called a zygote, Z-Y-G-O-T-E. Zygote is only present for about 45 minutes, and then it starts dividing very rapidly, undergoing mitosis and cytokinesis. Mitotic and cytokinetic divisions occur so quickly that there's no time for cell growth to occur. So you can have hundreds of cells and still not have the embryo any larger than the original oocytes. You guys know this from looking at what? Where have you seen evidence of this? Where the divisions occur so rapidly that the cells don't get bigger. You just end up with a whole cluster of very, very tiny cells. In lab, you looked at this. Through the microscope. What?
Fish.
Yeah, the whitefish blastodiscs. That's why those cells were so tiny. The cells in the other tissues of that fish, in the adult fish, are not going to be that small. The reason those cells were so tiny is mitosis and cytokinesis is occurring so rapidly, there was no time for cellular growth to occur between divisions. So you ended up with smaller and smaller cells as a result. None of these cells has a specific job yet. We just have a mass of cells. It is technically an embryo, but pregnancy still hasn't happened. For pregnancy to occur, that embryo has to be moved down through the uterine tube into the uterus. And as it drops down through the uterus, it just randomly kind of bounces along the walls. And that corona radiata, it becomes kind of spiky and sticky. If it manages to stick to the endometrium, they call that implantation. At that point, part of the embryo starts growing literally into the endometrium. And that forms the beginning of what will eventually be the placenta. When that happens, the embryo takes over production of estrogen and progesterone. By this point, that corpus luteum left in the ovary is starting to degenerate. The embryo itself, some of the tissues from the embryo take over that function and prevent more follicle-stimulating hormone from being released to initiate another ovulation. At this point, pregnancy has occurred. Without implantation, there's not going to be pregnancy. If the embryo doesn't implant, okay, gang, it's a tiny little thing about the size of a period at the end of one of the sentences in your textbook. It passes right on out through this external os of the cervix and out through the vagina and dies. And that happens more often than not. Implantation is a chancy thing. So pregnancy doesn't actually formally begin until implantation of the embryo occurs. It's not enough to produce the embryo. We have to get the embryo implanted in that endometrium of the uterine wall. All right. Now, a couple of things that people invariably ask about, so I'll kind of do a preemptive strike here. People ask, well, what about twins? What about twins? Well, there's two kinds of multiple births. There are maternal twins, in which case, during those first few divisions of the embryo, some of the cells become completely separated from one another. As it has not been determined what any of those cells are eventually going to become, each cluster of cells develops into a complete embryo, albeit a slightly smaller embryo than it would have been otherwise. These would be identical twins, or quite literally clones of one another. They are genetically identical. These are coming from the same zygote. The other variant on this would be what they call paternal twins. And this can occur in the event of multiple ovulations happening, where you have more than one secondary oocyte being released either by one ovary or both. Then that means there's more than one secondary oocyte that can be fertilized by different spermatozoa. As the spermatozoa are all genetically unique, and the secondary oocytes are all genetically unique. What you end up with is two embryos being produced at about the same time or more that are not genetically identical. Fact of the matter is, they don't even have to be the same gender. They don't have to look any more alike than any normal set of siblings would. And then both would have to, or however many there are, would have to independently implant. All right, so we've got two different variants on this. And if that doesn't make life complicated enough, both can happen at the same time. I actually knew somebody where this happened. This individual, I knew him years ago, had a wife that, well, first off, they decided they weren't going to have kids, which is, of course, the kiss of death. Second, when they got married, they weren't going to have kids. So they found this little girl they adopted. And adopting a kid, it even ensures pregnancy more. I don't know why, it's some kind of cosmic thing. But a year or so after they adopted the little girl, she's pregnant. She was a fairly large-framed individual. I never met her. She had to be, because she gave birth to triplets. Really big triplets. They averaged right at 7 pounds. She carried 21 pounds of baby. Okay,
divisions in one embryo, and then a completely separate embryo from a separate sperm and ova due to multiple ovulations, which they didn't know at that time she was doing. Most people don't. Okay, this guy, by training, he was a virologist, so he didn't really understand eukaryotic things all that well. And the assumption was made that there was a window of opportunity that didn't actually exist. So about 4 months later, she's pregnant again.
What?
We shared an office. And, you know, this guy, he only stood like about that tall, weighed maybe 90 pounds. And he looked like he was 15 years older than he was, I think mainly just due to the stress. Because, you know, they had a little girl and had 3 babies that they were having to assembly line diapers on. And I knew that she was pregnant, and he came in one day and he said, well, she went in for an examination. They picked up 2 heartbeats.
Oh my gosh.
And then a couple months later, when we did sonograms and everything, he came in and he said, Greg, I don't know what I'm going to do. Well, what's wrong? It wasn't so much the first group trip as he referred to as the wolfen. Or the idea of the twins, but the sonogram picked up 3 implanted embryos, twin boys and a girl.
Oh my God.
Okay, anytime you think your life is rough,
You know what I mean?
imagine 6 in diapers at the same time. Imagine having a house later on with 6 teenagers all wanting to drive. And then 6 going to college within a 2-year period. Yes, your lives could be much worse than they are now.
So they were only 16, about 16, how far apart were they?
Well, she was pregnant like 4 months after giving birth.
Oh my gosh, they're just barely over a year.
So, yeah, they were very
That's like
close to
14
each other in age.
Yeah.
Yeah.
That's
I
what
have a question about, so birth control pills would suppress the follicle stimulating hormone?
Well, but basically, it depends on the pill.
Okay.
Different birth control pills do different things, but a lot of them just increase estrogen concentration, which inhibits follicle stimulating hormone. Yeah.
Now, the intrauterine devices,
What they do is they create an environment that is physically, hypothetically, it's physically lethal to either the gametes or a new embryo.
so that's also done by hormone?
No, in many cases, it depends on the specific intrauterine device. Some of them is just by presence of certain kinds of metal ions.
Okay, and then what about a tubal ligation?
Oh, tubal ligation is where they take and cut the uterine tube and tie it off. So basically, fertilization can't occur, and the secondary oocyte just ends up breaking down and being reabsorbed.
Okay. So then why does menstruation still happen after a tubal ligation?
Because you're still going through all the same hormonal changes. Menstruation occurs when implantation doesn't happen. You get a drop in estrogen and progesterone because of the degradation of the corpus luteum. And you can't support that thickened endometrial lining, so some of it sloughs off. All of that's still going to happen, even with the tubal ligation,
So it
because those
doesn't-
hormones are being released into the blood.
Okay, all right, so it's not like
It's not like the ovaries have been removed. They've just tied off, essentially, the uterine tubes.
so the production of the secondary.
You're still going to have ovulation
happens.
occur, all of that's still gonna happen, it's just it can't go anywhere.
Okay.
So it ends up breaking down, and being reabsorbed. Yeah,
So when the women then like stops off, you know, when
Uh-huh.
women are having their menstruals, so I know, so is the, not trying to works, but is the blood like,
Well,
just because the endometrium
the-
is highly vascularized?
it's highly vascularized, and you're actually sloughing off those extra capillaries and everything as well. So there is some blood that's lost. Yeah,
Okay, sorry, I have a friend, she's having triplets, but they did it like, I cannot remember the name of the stupid thing, when they mix it up in the petri dish and then she had to put it in vitro. Thank you, that one. Okay, so when they're mixing it up already, so then they already have the zygote going. Okay, so now it's up in there, shouldn't it have just fallen and implanted? Cause they stuck in two, because they figured if one fails, they get one. She's having triplets because one of the zygotes, like, went apart, she's having identical twins
Yeah.
now. But then, I don't know, I'm confused, if it's already mixed together, why does it fall apart again if it's already an embryo? So then,
it's during the first few divisions.
okay,
The cells, instead of staying stuck together, completely separate. And then each cluster of cells develops into a complete embryo.
okay. So
That's
it's
how
just
that
those
works.
cell clusters, okay.
So what she's gonna have is a situation very like this guy I was telling you,
Mhm.
where she's gonna have identical twins and then a third sibling that would be essentially maternal.
Okay.
Now, when people take, when women in particular are taking fertility drugs because of some fertility issue, one of the side effects of that is it very commonly causes multiple ovulations in people that don't already do that, which is why multiple births are more common when people are taking, when women are taking fertility drugs. It wouldn't work that way with men, because you still have the same number of secondary oocytes to work with. You know, you only have typically one being released at a time. But in women, as a result of the